Hello, welcome to the demola, and today's lesson will be covering mitosis, another very important foundational topic in A-level biology, where we'll be uncovering the infamous PMAT. No time to waste, let's get into it.
Okay, so even before getting into mitosis, we need to talk about cell cycle. Now, your cell cycle consists of multiple stages, but they're split broadly into interphase and then mitosis, right? So in interphase, you've got gap phase 1, DNA synthesis, gap phase 2. And then in mitosis, you have the actual process of mitosis and then cytokinesis afterwards. And we're going to get a lot into like what's happening in each of these stages. But yeah, I should have actually started with the definition of the cell cycle, which you can see on the screen. But it's a sequence of events that occur during cell division. Yeah? Cool. So, interphase. This takes about 90% of the cell cycle. So just know it's a very large portion of it. And like we said, we have the gap phases, but really, these are the stages where you see growth. So your cell is going to get larger. It's going to replicate organelles because we're getting ready to split, right? In G1, you're going to make proteins for DNA replication. G2 for mitosis. Why is that? So the S phase is literally just DNA replication. So like, we've done a video on this channel titled DNA structure and replication. So if you look at that video, then you'll be able to get like the steps of DNA replication, etc. But just knowing that S phase, we have DNA replication occurring. So if we want to just say it plainly, what's happening? G1, your cell is growing. It's creating new organelles, right? And then DNA replication happens. So we double the amount of DNA that we have. Then we're going to go into G2, which is when we're really preparing for mitosis to happen. And that is really the crux of interphase. One thing I did want to mention as well, not too relevant for A-levels to be honest, maybe a bit beyond that, but you do have checkpoints in your cell cycle. So these are like stages where we're just going to do some checks, make sure that there hasn't been any errors in the DNA replication, make sure that there's nothing wrong with the cell. This happens when you're in G1 going to S phase, and when you're in G2 going into M phase, and it also happens during mitosis as well. So yeah, you're checking for errors mainly because you don't want cancer to happen. So it's mainly to prevent cancers. And some of the cells that are like, you've seen they have an error, that's a dangerous cell. They're going to G0, which is like you exit the whole cell cycle. So that's not involved. Some of them can get back in, but they just exit the cell cycle. But don't worry too much about that. That's very much an additional point. Okay, now we're actually going to talk about mitosis now. And really, things that you need to remember is we're producing two identical daughter cells. So, what do we mean identical? We mean that they have exactly the same DNA. So exactly the same genes, chromosomes, whatever, they are genetically identical. Yeah? Mitosis does only happen in somatic cells. So somatic cells is basically every single cell in your body, except from the gametes. I've always been cooked for pronouncing it wrong. Except for the gametes. Yeah? So those are the cells that are going to undergo mitosis. And the real question is, why do we do mitosis? There's always a reason. And mainly it's for like growth and repair. So as you're getting older, say that you like damage your arm, you scrape your arm or something, you need to regenerate the skin. This is why we're doing mitosis. So the skin thing is more about the replacement of the dead cells. Yeah. Can also be like in the hair as well. So your hair cells die, get replaced. Asexual reproduction as well in some bacteria, they'll use mitosis. So these are indications for why we do mitosis. Cool. Mitosis occurs in 4 stages, and as I said in the intro, we're mainly going to go by the acronym PMAT. So we're going to get into each stages, but you mainly use PMAT for your stages of mitosis. Now, the first stage that we're going to talk about today is prophase. So in prophase, what's happening? Well, we've had interphase, yeah, and now your chromosomes are going to condense. What does that mean? Chromosome condensation just means that they're going to become shorter and thicker. Yeah, literally that. They become shorter and thicker. And now we can actually see them under an electron microscope. Yeah? What else happens? Nucleolus disappears, the nuclear envelope dissolves, and the centrioles replicate and move to opposite poles. So I'm just going to quickly highlight some stuff. These are your centrioles. And then those are your chromosomes, or we'll call them sister chromatids at this point. So you can see them in that cell. After prophase, we're going to move on to metaphase. So, metaphase, what's happening is your centrioles release these spindle fibres, and they're going to attach at the centromere. So, if I quickly draw out one of our chromosomes, sorry about that. If I quickly draw out one of our chromosomes big here, the centromere is just in the middle where they connect. Cool. So I'll just circle that as well. That's the centromere. Now, spindle fibres attach to the sister chromates at the centromeres, and they start to line them up along the equator or the middle of the cell. So you can see that here in red, the spindle fibres attaching to the centromere, and they're released by the centriole. They attach to the sister chromatids and they line them up across the equator of the cell. Following that, we're going to have anaphase. So in anaphase, what's happening is those spindle fibers start to shorten. And be really, really intentional with your vocabulary. So we're saying shorten, we're not saying contract, we're saying they shorten. The spindles shorten because they aren't a muscle. Now, the spindles shorten and they pull the sister chromatids to the opposite poles. So you can see that centromere gets split, right? And your sister chromatid now, it's in two separate parts. They move to opposite poles of the cell. So they're moving to the opposite sides because the spindles are shortening, as you can see in the diagram here. Now, last phase of mitosis is telophase. And in telophase, we're literally kind of doing the opposite of what happened in prophase. So like your chromosomes decondense, can no longer see them. Nuclear envelope is reappearing, nucleolus reappears, your centrioles dissolve, and you kind of get like this. So the cell membrane is also going to pinch inwards so that we can do the next stage. But you can see here, we've got a cell that characteristically has 2 nuclei in it. Yeah, that's an indication that it's in telophase. But you can see that telophase, we're literally doing the opposite of prophase because in cytokinesis, which isn't part of mitosis, but it's part of the mitotic phase of cell division. But cytokinesis is when your cell membrane splits in half. So we have the pinching and it just keeps going all the way along the middle. So like if I added this here, it would keep just going and going and going and going and going until the cell splits into 2. And when it splits into 2, you know, in cytokinesis, we have 2 genetically identical diploid. We haven't done this terminology, but diploid means it has like a normal number of chromosomes. So like 2n or 46 chromosomes in a human. And then you've got 2 cells. Beautiful, cytokinesis. I did want to show this diagram as well. So this is a bit important for exams as well. DNA content in different stages of mitosis. So we're looking at one cell. Yeah, this is just one cell. And we're looking at its DNA content across the cell cycle. So in G1, it's at 2n or 46 chromosomes in a human. In the S phase, DNA replication is happening. So we're doubling the amount of chromosomes we're doubling the amount of DNA that we have. The chromosome stays the same, but we double the amount of DNA that we have. So if we double the amount of DNA that we have, we're at 4n, right? Now, G2 is going to happen. And then in the mitotic phase, because cytokinesis is going to happen, that one cell is going to split into 2. And when it splits into 2, both of the nuclei have the original number of chromosomes. So we go back to that 2n that we had in G1. I hope that makes sense. So just remember, you're following this graph with DNA content. When we do meiosis in a later video, we'll talk about how that is different as well. But just kind of remember this diagram here. And I believe it's question time now. So we've got questions here to do. First one, you're looking at metaphase in mitosis, drawing a diagram of a chromosome, and then also describing what happens. The next one, that graph we just talked about, I just want you to describe what is happening in stage 4. So pause the video, have a go at these. Okay, so having a look at this, like we said, what does the chromosome actually look like in metaphase? Well, we said that there's sister chromatids and are joined at a centromere. So literally, this is kind of what we wanted to see. And you can have the labels there, you can see them here. So you have one chromatid that's labeled there. There's the other chromatid that's unlabeled. The centromere is where they meet in the middle. So that is what your chromosome is going to look like. And then also what we need to say is during metaphase, you're lining the chromosomes or the sister chromatids along the equator, right? So there along the equator, the spindle fibers form or they attach to the centromere. Now, I don't know why it's not saying do not accept chromatids. That is a bit odd to me because it is sister chromatids. So I would kind of ignore this bit of the mark scheme, to be honest, because sister chromatids should be fine for this part. Cool. Now, the next question as well. So we said what's happening in stage 4. But you can see that's the mitotic phase. So really what we're trying to get at is cytokinesis is what makes it go down back to 2n. Yeah. So the DNA content is halving. That's an observation. Cytokinesis is happening. You're forming the 2 daughter cells. Hope that all makes sense. Okay, and now we're on to the A-star tips. So stages of the cell cycle is pivotal. Remember the stages of the cell cycle, interphase, and your mitotic phase. And recording the stages of mitosis and telling your examiner what is happening during those stages is also really, really, really important. So remember the PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis after. Cool. Stages of mitosis on an electron micrograph. We didn't cover this during the actual lesson, but it's really, really important. So you can see here, if we start with prophase, you can see that you've got your cell, you can clearly see the chromosomes. So in a normal cell, like this one here, you can't see the chromosomes. Here you can. Now, metaphase, you can see they're lined up along the equator. So again, we're just going to highlight, you can see they're lined up along the equator there. In anaphase, you can see here that they're splitting off. Yeah, this one is interphase. So again, just like that one, you can't really see. You can't see the chromosomes individually. Yeah, and then if we had to go for something in telophase, there might not be one here, but maybe this one arguably is in telophase. You can see like two distinct nuclei, essentially. That's kind of what we're going for when we're saying telophase. Cool. And then what not to do. We haven't covered meiosis yet, but it does get confused a lot because they sound really similar. So props to whoever did this, named these things in biology. They sound similar, so they do get confused quite a bit. But remembering mitosis versus meiosis, we'll do another video on meiosis later. So this is the chromatid versus chromosome. It's important to know when the examiner wants you to say which one. So normally in mitosis, when you're talking about the actual process, we're going to be saying sister chromatids. So that's in metaphase and anaphase, right? Otherwise, you are probably saying chromosome. And then neglecting interphase, I think it does get a bit neglected. Like people won't know what's happening during the gap phases or the S phase because they kind of see mitosis as the big topic because you need to know more in detail about what's happening. But remember, interphase is 90% of that cell cycle. So very, very important to know what's happening in interphase. Hope that's helped. So if you found the video useful, please like, subscribe to the channel as well if it was really good. Drop a comment below on any questions you have. I know especially that thing with the mark scheme might have been a bit confusing, so I'm happy to clarify if need be. But yeah, any questions, put them in the comments below. We've also got the TikTok and the Instagram, both at demola. Links will be in the description below, and I'll catch you in the next one. Bye for now.